Welcome everyone, one more time, not the last time hopefully. We're going to have an internal kind of display today, and welcome everyone, and also the ones online later recording or watching the recording. We're going to try YouTube now. We set up a channel and it's going to be the first time, so bear with us. It's going to be a fun ride. And we're going to have a very interesting talk by Éléa today from the newly created project GLIOMETER that came out of ideas from the Tumor project. And I'm very happy to let you present, and we'll happy to take the questions after.
My name is Éléa. I'm working actually here with the Oro project, and this project is kind of part of ORAO, but not entirely part of ORAO, and we will understand why. Basically, for you who don't know, which I guess is not many, working on the ORAO project, in which basically we're using mirror parametry to visualize brain tumor margins during surgeries. There are many problems with seeing the margins of the tumor during surgery, and there are no tools that really allow to detect and to see the borders of the tumor at the same time that allows to visualize the fiber tracts. And we're using actually mirror parametry to solve this issue. Why we're using mirror parametry is because the brain fibers that you can see here in white are creating optical anisotropy, and basically with the optical anisotropy, it's a measurable parametric property that we can see with our instrument. Why is it important? Because when you have tumor, and when tumor is growing, it disrupts the organization that we have in the brain fibers, and this also disrupts the optical anisotropy that is due to tumor growth. And basically, if we see the absence of optical anisotropy or order in the tissue, then we would assume that this region is tumor. We had, 2 years ago, we published this paper in TMI, which basically was the first proof that there is actually a contrast that we see using our technique for brain tumors. Here what you can see is actually a tumor-free sample. We have different parameters. I will not go too much into details, but basically in the retardants, we have higher values in the white matter, and we have low noise in the azimuth of the optical axis, which basically represents the orientation of the fibers in the tissue. What we saw in most of the tumor samples is that actually we have a significant drop in the linear retardance when we have tumor tissue, and there is much more noise in the azimuth of the optical axis than what we have for our tumor-free tissue, which makes sense because we have disruption of the organization of the tissue microstructure. We quantified the values for different areas, and basically in green here you have healthy tissue, and in red you have tumor tissue, and we found two parameters which gave very good contrast, which were linear retardance and the azimuth local variability. So this was a good first step. There was, however, a problem with this approach, is that we're basically quantifying the labels were made on histology, and the problem was that we were quantifying and creating categories where there shouldn't be categories. Basically what we did is that we put what was below 10% as healthy tissue or tumor-free, what had between 10 and 70% of tumor cells as infiltration zone, and what had more as tumor center, more than 70% of tumor cells. But this create boundaries because something that looks like 60% of tumor cells, something that has 80% of tumor cell is not that different, and we're creating basically a boundary at 70% where it's actually continuous variable that we're looking at. So we had an idea, and this caused a problem, This caused a problem because I'm going to try to take you on a game and basically play the role of pathologist, and we will try to see what is the problem together. This is an example of an H&E patch of, I'm pretty sure everything I included is astrocytoma. This is a patient that had an astrocytoma. In this patch, there are actually 55.52% tumor cells. This was quantified using immunohistochemistry. It's pretty obvious. It's pretty obvious if we look at this slide here that there are more cells and probably more tumor cells. Actually, there are more tumor cells because we have 94% of tumor cells in this patch. Here, it's also pretty obvious that there are less cells, there are probably less tumor cells. This looks more like normal tissue, and we have only 17% of tumor cells. Sometimes it's less easy. If someone had to guess if this is actually close to the boundary, this 70% boundary. If we ask any pathologist, probably half of them would say this is below 70%, half of them would say it's above 70%. This one actually is slightly lower. And probably the same with this patch. It's also very hard, even with training, to say if it's actually below or above this threshold. And actually here we are slightly above the threshold. But as you can see, there are not so many differences. There are actually even a bit more cells in this region than you have in this region. And it's because most of the more cells are positive, so here negative, so here they're labeled in green, compared to what we have in the other ones. We have less cells in general, but most of the cells are actually tumor cells. And also, what we see is that if we use the classification as we have it now, so basically putting everything that is between 10 and 70% of cells together, we would put these two kind of tissue together, where actually they look completely different. Here we have a few tumor cells here and there, but most of them are healthy cells compared to this. And we're basically reducing the amount of information that we're using, and we cannot capture completely the differences that we have in the microstructure of the tissue. This idea, the first idea was to use it for the ORAO project, because to validate the findings that we have with ORAO, we cannot rely on estimates of this tumor cell content and on these broad classes. On top of this, we know, and it's been shown in literature, that the estimation by pathologists is subjective and there is a high inter-observer variability. So there was a need to standardize the method that we were using. So to solve this problem and to get estimates directly, we propose to use foundation models. I don't know how many of you know foundation models, but basically the idea is that these are very large models, pre-trained models that basically learn how to represent, especially in pathology, learn how to represent complex images as a set of feature vectors that represents the best of the tissue and then can be used in classes, in downstream tasks. For example, you can use it to classify tissue, you can use it as a segmentation network, you can for example, determine what subtype of tumor you have in certain histological slides, and there is a big trend in pathology for foundation models. Basically, GLIOMETER extracts these deep features that are generated by the foundation models from routine H&E slides and predict patch-wise tumor cell content values. We also realized that this was not only about ORAO, but this could have way bigger implications in the future, because basically this could be a quantitative framework for high-grade glioma, and this could solve an important issue in routine neuropathology, especially current neuropathology. Basically, nowadays the neuropathological diagnosis that you have is algorithmic and it requires high quality molecular data. Typically, we have they need to use they need to use samples and tissue regions where you have more than 70% of tumor cell content. Why? Is because nowadays they use especially methylation data, and this test requires to have more than 70% of tumor cells to ensure that what you are seeing and the results that you are getting are reliable and can be used for the classification. Nowadays, especially, probably it was not the case 10, 15 years ago, where they relied only on the morphology of the cells, but now they rely also quite highly on the molecular data and on specific alterations, and sometimes alteration such as the deletion of CDK and 2A or B, and allows by itself to characterize a tumor as an astrocytoma grade 4 even when you don't have necrosis and microvascular proliferation, which basically show that these two information, the molecular data and the morphology of the tissue, are used together for a current diagnosis in neuropathology. Basically, at the moment, the pathologists are showing where in the slide you have more than 70% of tumor cells, which can then be used for the molecular assays. But this is, of course, dependent on the pathologist, and it takes time, and you need to have someone who is trained to recognize these tumor cells. And we also thought that GLIOMETER could basically replace this information, this estimation that is used for triage, and make it more standardized and data-driven. This would also be useful because in pathology, modern neuropathology, you have, most cells, especially in gliomas, you don't have a marker that marks all tumor cells. When you have IDH mutation, then we can stain specifically for these tumor cells, but for glioblastomas, there is no way to stain only tumor cells, except in some specific cases if you have a specific mutation, where we don't have a staining that allows to get an estimation and to see only tumor cells. So this would solve issues also for routine diagnostic in neuropathology. So this is the framework that I've been developing. There are a lot of information. We're going to go through each one of them like piece by piece. Basically, it starts with, we have pairs of H&E and immunohistochemistry. We're using only, at the moment, IDH mutant tumors because there is a staining that only stains tumor cells, so we can get the ground truth and then base our predictions on. I will come back to it. But basically, we start with paired H&E and immunohistochemistry that we aligned using a software that was developed a few years ago, which basically aligns using first rigid registration, so basically just rotating and doing an affine transformation on the immunohistochemistry, and then we have a non-rigid registration, which matches more specifically the small details that you have in the different slides. Starting from these two outputs, we extract patches in both the H&E and the immunohistochemistry. These patches are located in the exact same region, meaning that the tissue that we see here is the same as the tissue that we see here, and then this allows to generate the ground truth. There was a strict quality control on the patches that we were using or removing. This, you have an example of 3 patches that we kept for the training and testing of the models. We removed patches which contain necrotic tissue because there were no cells and there were not really a point in using these patches anyways. If most of at least 15% of the patch was empty, to remove patches where we had less information, and it would also make the ground truth less reliable. Patches where we had high quantity of blood, because then we would have a lot of blood cells also, which could contaminate the ground truth. And also because we used a non-rigid registration, there were registration artifacts, so these slides with registration artifacts, these patches with registration artifacts were also removed from further analysis. One very important thing is that we're using a quantitative ground truth that is automatically generated and that is reliably generated. Basically, how it works is that we take the patches that we saw before, the immunohistochemistry patch. We have cell counting and classification of the cell that is based on QPath, where basically it finds the cell and then determines based on there is a built-in tool in QPath that allows you to generate very small machine learning models which perform good enough for this kind of tasks, where basically we just have here in red the cells that are marked as positive, so that are tumor cells, and then in green you have the negative cells, which are just normal cells, such as blood cells or white cells that are just here, but are not tumor cells. And basically, we take it for the patch that we have in the middle, as mentioned before. For each patch, we have the center patch and a more global patch to also have some context about what's happening around in the tissue, because maybe the region just surrounding the patch that we have has some different information, and we can kind of use more information also about the context. So coming back to this, then we take what's in the center patch, and we also include information about the patches that are around it. to try to smoothen and improve a bit the ground truth that we were using. And then we basically using a mean of what we have in the center and what we have on the side to get the final regression target using a weighted mean, putting more weight in what we have in the center than the surrounding patches. There were problems, especially because we have differences in the staining between the different slides, between the different patients. And in the beginning, if we used simple intensity thresholds, it didn't work because we have differences between the patients, we have differences between the batch. So yeah, as I mentioned, we used the QPath cell classifier, which allows to get a classification that is more reliable and robust, and that was validated to some extent so far with new pathologists, and that will be validated to more extent before we can publish this study. Here you have example of 3 patches that we are using. Here you have one patch which has tumor cell of 73.6%, where you have more tumor cells, and then you can see a bit of what's happening. And even sometimes when we have more cells, it doesn't necessarily mean that we have more tumor cells, because most of these cells can actually be non-tumor cells. And yeah, just then how does the training and the inference work? Basically, we take the H&E patch, not against the H&E patch that we feed into the vision transformers. That gives the feature vector. So typically it's either 716 or 1500 vector input vector that we then put into a feature-based predictor, which basically then is just a regression that uses as input the outputs from the vision transformers and predicts the regression target, so a continuous value between 0 and 1. We used the most recent vision transformers. There are a lot of vision transformers. They're always improving, especially in pathology. We used the most recent one that like a few months ago, which were these 4, and we benchmarked and see if there were differences and which one was performing the best. And we evaluated 2 tasks. So there was a regression task in which we predicted the tumor cell content of the patch, so basically this value between 0 and 1. And then there was also a classification task in which we predicted the patches which have a value higher than 70%. And so that would be the task that would then be applied for the neuropathological diagnosis. We trained the model so far on the subset of the data that we have. We used 15 whole slide images, and we have about 26,000 patches, and we achieved very great performance with basically all of the foundation models that we use. We got a mean average error of 6%, which is great, and I mean the correlation coefficients, both Pearson and Spearman, are really good also. Here, the AUC and precision, they're only based for patches which are between 60 and 80%, which is basically this hard zone. It's quite obvious when there is more than 90% of tumor cells, or when it's less than 50% of tumor cells. But what we're really interested in, what is really hard to characterize, is when we're in between this region where you have 60 to 80% of tumor cells. And even for these hard patches, we still have good metrics with AUC around 0.8 and F1 score 0.77. I guess this could be improved training on more data, but at the moment it's already good results. Now, everything that I showed is in IDH mutant tumors that we're using for training and for testing. We also tried to apply it directly to some glioblastomas. With glioblastomas, as I mentioned before, there is no universal marker that stains tumor cells. You can, if you have some specific subtypes of glioblastomas, for example, if you have p53 overexpression. So that's the samples we use to test our method also on GBMs. As you can see, the performance dropped. So we got from 6% and a mean average error of 6% to 13%, which it's minus 8%, and F1 score also reduced. So I tried to also use like to use some of the IDH wild type for training, so kind of co-train on these two, on both IDH mutant and IDH wild type. And this increased a lot the performance, and we got basically the same performance that we had in the IDH wild type and IDH mutant tumors, where it basically went down when it's co-trained to 6% mean average error. And the F1 score increased a bit, although it's a bit less than in the IDH mutant tumors. And importantly, we were able to increase the performance, especially mean average error, on IDH wild type tumor, but it didn't alter the performance on IDH mutant tumors, because basically, as you can see here, the metrics are basically the same, meaning that we can increase the performance in IDH wild type tumor fairly easily by co-training on a very small subset of data, and this doesn't alter the performance on the IDH mutant tumors on the samples on which it worked previously. Finally, just to mention how, like, the performance, how long would it take, how long does it take at the moment, for a whole slide, the full inference to get something that looks like this, as like a map of tumor cell content, takes about slightly more than a minute for a whole slide image, which is way less than it takes to generate these images, which is a good thing, because it means that in theory it could be applied in diagnosis, and we get pretty consistent map with the ground truth. This is the ground truth that was generated from the IDH1 staining, and as you can see, we have something that more or less makes sense, in which we had higher values of tumor cell content here and in this region here than we have in this other region here. Finally, just to conclude, basically what started as a tool to calibrate our parametric system became a solution to a systematic diagnostic bottleneck in neuropathology. With this tool, we can provide automated tumor cell prediction with great correlation to the biological ground truth. It generalizes across heterogeneous tumor types. It can adapt to new unseen data with minimal co-training with only a few whole slide images. And this provides a solution to facilitate and standardize the implementation of the new classification in neuropathology on a routine clinical basis. With this, I want to thank all the people in this project, and yeah.
That's on me.
Can you please show the previous picture? Is this from Mykonos?
Yeah, yes. Of course.
I would have a question for both of you.
Mm-hmm.
Can you go back 2 slides? 3 slides. Next one. Yeah. So all the statistics you've shown are basically on a patch level. But in this image, you see the context, right? These patches are really tiny by comparison with the size of the whole slide image.
Mm-hmm.
And I wonder about the, especially when you talk about kind of a mean average error of, I don't know, 6% or something like that. When you're probably the interesting statistics are not patch by patch, but something less granular than that.
Yeah.
And if you average over larger spatial domains, or do those kind of deviations average out and you actually kind of get better results? And what's the granularity on which these kinds of, in pathology, what's the granularity on which these kind of results would be interesting?
I mean, to answer like the first part of the question about like this is a slide that I think I made 3 months ago. Since then, the size of the patch increased a bit, so it would be more average, kind of.
Mm-hmm.
But as you can see, also the ground truth it's not exactly like it's really following what we would see in the ground truth. Probably it would be what could be. I mean, I don't know what could be useful probably for a pathologist would be to have bigger regions,
mm-hmm.
which says where you have more than 70% of tumor cells. Of course, we wouldn't want like a small box saying here in this small box you need to take this, in this small box you need to take it, but more global and maybe probably some average across the. And I guess I guess there could also be some, you could also check the outputs of the model and also check if it's consistent one with the other, because if you go from 0 to 100%, there is also something wrong going on. And I mean, maybe there could be a penalty in the model when we train the model to ensure that this doesn't happen.
Yeah.
But for most of what I saw, I never had something where we went from 0 to 100% and it was more or less continuous everywhere. Here it's a bit different because here we have necrotic tissue here, but otherwise in the other regions where we have tissue, it's more or less consistent, and we don't have very big differences between two neighboring patches.
And if you think, I mean, you have to ask yourself, what is the aim? I mean, GLIOMETER can serve multiple aims, right? And if one of those is, like Éléa said, to assess the tumor cell content, for example, in order to perform molecular analysis, right? And in order to have such a successful analysis for the final classification of the tumor, then the technicians usually they do biopsy stances, small biopsies of the paraffin block, and usually it's around, I don't know, 0.3 centimeter in the diameter.
Okay.
So you would like to have in this kind of the diameter the information of what's the tumor cell content.
Yeah. But the important thing is that the overall tumor cell content is higher than the minimum threshold.
Yes.
And if you have, with this, maybe there's variation across the patches, but if you average across it, you see, okay, here is the area that's most likely to contain the highest.
You want to have the area with the highest tumor cell content.
Yeah.
Yes, you want to avoid areas that have less than 70 or less than 50, let's say. And this was the biggest challenge to, once we're setting these classifications, already also in the frame of the Éléa group. And I mean, sure, you have less than 10%, the brain tissue morphologically looks almost normal. If you're in the tumor center, you have more than 70% of tumor cell content, it's also very clear. But what happens in between? This was always the burning question for us. You're between 10 to 20 until 70. In the beginning, we had like low-level infiltration, high-level infiltration, but to find the gradient was really difficult. And then we just put one infiltration class for high and low level.
Yeah.
But the aim is always to get most of the most hypercellular area where you have most tumor cells. And for that, sure, the size is important. If the patch is really minimal and then you're next to a low-level infiltration area and you make a patch from, it's not going to help the molecular analysis at the end.
Yeah, maybe to amend this, you could include a probability in some way, how secure the model is in predicting what is here, or in segmenting the tissue correctly. And then you have two kind of modalities in one image. I don't know how to display that.
I mean, if you know what is the size that the size of the patch that you would use for neopatology, you could just take the patch which has the higher tumor cell content in this 0.3 centimeters region, and maybe take first the top 3 matches or something like this to guide the analysis that would be then performed.
But right now these patches don't overlap, right? It's just
No, they don't overlap. No.
And if you had bigger patches and you wanted to be robust, I think you would want them to overlap, right? So that you avoid that you have you split the highest tumor cell density area in half.
But I mean, probably you could still take small patches. I mean, the patches I'm using now, they're 4 times bigger than this, but still they I don't know exactly. Probably they're I think they're 128 micrometers. The patches I'm using in size. So I mean, it's still rather small, but you could increase the size of the patch at the end of the day if you want. Then the only thing is, I don't know until how big you can increase it and run it in a standard GPU.
No, I think the smaller patches seem to be slightly bigger, but I think the smaller patches.
Yeah, I mean, anyways, the main thing is all these foundation models. The first step that they use in the forward parse is basically to resize everything to a 2 to 4. So it doesn't matter. You can take a patch bigger. The first thing that they do is resize everything to 2 to 4 times 2 to 4. So there is not really a point in bigger.
I would think. If you have to think about the use cases and how we want to put this on a paper so that it gets nicely published. And so to to recognize on the HNE slide the hypercellular glioblastoma area, to annotate it so that the technician can do the biopsy and do the molecular analysis is not very difficult. Where the problem would arise would be, for example, if you have a low-grade glioma case, which would maybe look like almost normal brain tissue, slightly hypercellular, and then you don't know, do I have more tumor cells, meaning more chances to find the mutation in this area of the tissue or in a different area of the tissue? In such cases, you would need something like a gliometer to tell you, in this area, it's a low-grade glioma, the tumor cell content is not very high anyway, but in this area you have 20% and not 5%, so you have more chances to be able to find a mutation or a fusion, so more chances to make a diagnosis. This probably will come as a comment. Maybe we have to think if we have to find a way to comment on it already from the first submission. And the second part would be, for glioblastoma, as I said, to find a hypercellular high-level tumor area is not difficult, but then you can argue that, you know, it can also be used. Now, for example, technicians come to the neuropathologist and say, please annotate the slide for me, takes time and so on. If it's an automated thing that runs in the lab and the technician can put a slide by himself, see the tumor cell content, I do a biopsy in this area, I don't have to go to the doctor anymore, we save time and so on. This would be an argument for the glioblastomas, for example.
I want to hook on the comment on the GPU. What inference testing do you do on your local laptop? Or?
No, this is on 4090.
No.
But just on
you can ask your market to run it on the Holoscan.
Yeah, probably would. Yeah, probably would be a bit faster, but yeah, I mean, yeah, it would be faster.
Yeah, for sure.
But I mean, this is just tested like on the just consumer grade GPU.
Yeah,
just,
of course.
Stating basically that any person with like decent GPU can run something like this.
Yeah, the minute is fine in a lab setting. Doesn't have to be real time. Would be nice, but.
Yeah.
And then the other thing I was wondering, you mentioned the co-training with the IDH mutant and the IDH wild type.
Yeah.
Why do you think that it has such a big impact on the glioblastoma ones, but not on the other side? Just because the sample size for training is bigger, or?
No, from what I saw, I didn't include it here. But from what I remember, it looks like a calibration problem. When I applied it on the GBM, like the IDH mutant region, it identified correctly which regions are low and high tumor cell content. I mean, yeah, as it should. But it looked like in the where you had like lower regions, like lower tumor cell content, it kind of overestimated it compared to what you have in IDH mutant tumors, and that was mostly the reason. Basically, it was always in the direction of overestimating, and it was overestimating the tumor cell content by 10 or 15%.
Yeah.
And when you changed and when you co-trained, probably just the model learned that these tumors look a bit different, and then you should.
Okay. So the range.
Kind of recalibrating the instrument for this specific tumor type.
Okay.
And probably also if we train with more samples to start with, because now we train on 15 whole side images, which is not a lot for these kind of things. If we train with more, then the model will get more robust and more. It will generalize better to start with in the first. That's the next step.
We haven't trained on any low grades, right?
No.
Which would be nice for the first submission, even as an appendix. We have to comment somehow about it. These are the most difficult case to recognize on HNE, so this is where the more interest lies. Very nice.
Cool. More comments, questions?
I like the name.
Yeah, it was a great idea.
I love to play. All right. Then thank you very much. We will close the formal format.